Hello, interstellar everyone. Welcome to class 11 biology. In this video on chapter 13, photosynthesis in higher plants, we are going to discuss about C4 pathway. Right. In our previous video, we have discussed about C3 pathway called Calvin cycle. Imba dingye denga zu video nanglola C3 pathway ngal jaimba Calvin cycle. Dinga zu kharikan shisha gore C3 pathway the C4 pathway la di kharikan shisha gore la imba na. If you remember, I told you what is the first product formed after carbon dioxide is fixed. Carbon dioxide plant ki subu nanglola ya fixed chewa ta product thangbu di khari form chigido digala shisha ni plant la rigmanda ni yores la so C3 plant ta C4 plant. C3 plant nanglola, what happens when the carbon dioxide is fixed? The first product is a 3 carbon organic acid, 3 carbon containing organic acid called as phosphoglyceric acid, PGA form chigiwa. And C4 pathway nanglola kharchi gore C4 plant nanglola carbon dioxide ya nanglola thewata and carbon shi yogi acid form chigor. So we will look at the C4 pathway and we will see how C4 pathway following plants or C4 plants, how C4 plants are different from C3 plants. C4 plants ta C3 plants ni parala khewa khardu di ko chwe cherda. Ranga jo C4 pathway kan jang du zambala khare ha ko gore C3 plant ta C4 plant nanglola kuni ni parala khare khewa do. Dinga ju ko chwe gore. Okay, so I hope you understood the objective thanda ju di video the objective dikhna ko chwe share. Ta see the C4 pathway la zambala khalla gore. So which plants they follow this C4 pathway? Plants adapted to dry tropical places, they have got C4 pathway. Tishin saja kambo ani chawa chawo yosa sajala nima shukchumbu pesharsa sajala ani plants ge khari pathway kandi follow chigor la na C4 pathway they follow chigor, not the C3 pathway, right? And we will understand why khajin follow chigumedi chelo ko suyurta. Now these C4 plants, right, how they differ from C3 plants is these C4 plants they have got special type of leaf anatomy. C4 plants, the plants which follow C4 pathway, right, these plants they have got special type of leaf anatomy. Loma nanglo gita kejo jomarwe suk jomarwe di mandajur, they have got special type of leaf anatomy. Leaf cross section tagrta, which makes them more tolerant to higher temperature, right, and they have got greater productivity. C4 plants nanglola ani khonso special leaf anatomy yobe kengi ani khonso khariore higher temperature nanglo khonso yaga survive chetu wor nima ki intensity shukchumbu yomine sunlight intensity shukchumbu yobi mene khonso survive chetu wor even sunlight shukchumbu be yobi mene khonso khaju wor dizibabe ki khonso photosynthesis ke yaga chi wor. C3 plants nanglola, what will happen if the sunlight is too strong, if the sunlight intensity is too strong, C3 plants they get damaged. Chlorophyll thula du gor la so dinalo khar C4 pathway nanglola C4 plants nanglola roa. These plants they are adapted to survive in these dry tropical places, right? So they have got special leaf anatomy. So if you look at the vertical sections of leaves of a C3 plant and C4 plant, C3 plant gi loma cross section ji vertical section na tubyo la gorda loma leble ji khaptu gorma tubo imba na vertical section. If you take a vertical section of the leaves of a C3 plant and compare it with C4 plant, right? If you to compare chewe imba na ani kiranzo kuni ni para ge kewa de gotsur anatomy ki kewa khari yomedi ani C4 plant la special leaf anatomy condition to di kiranzo gotsur ta kiranzo thonan khari late go gore. You have to look at the differences in mesophyll cell and the vascular bundle sheath cells. Ata di ngechela remuna ta wende remudina ta da kiranzo silber. This is a C3 plant and this is a C4 plant. C3 plant ta C4 plant ni para gita leaf tumni kewa ditta wa imainar da. You will find that these are vascular bundles, right? These are vascular tissues. The cow roast vascular tissues. So let's say this is a rice, right? This is a rice plant. Rice plant dikhare C3 plant. So dikhe si de gaji zishin jin bacha sha koi loma jin bacha sha ro ani loma din nalo kinas di cow do den sama khare vascular tissue na khare transportation nalo ro ba chinge di ro chuda ni khala na din ya xylem the film din sula. So these are vascular tissues. Vascular tissues ki chilo la khardo changush to nezula ngazu khala ore bundle sheath cells la wor, right? So vascular tissue is covered by bundle sheath cells. Ani sur gi cell dige sama khare all these cells they are mesophyll cells. adi C3 plant gaji ki leaf anatomy dikhran se uchung so. Now look at the C4 plant leaf anatomy. C4 plant ki leaf anatomy la tawa imba na. They also have got vascular tissue. Now outside the vascular tissue, look at the bundle sheath cells. Bundle sheath cells di nangshi mandawaj khari ro delo thuga yore. Sometimes it is made up of several layers of bundle sheath cells. Vascular tissue ki chilo la tumgyosh midwe. The vascular bundle ki chilo la tumgyosh toa. Dila ngazu khali bundle sheath cell lagro. Bundle sheath cell di che yore and samsam kharchi gore. It forms multiple layers of cells. Yoshung so. Ta di chikbu marda. Di chilo la doni mesophyll cell la tawa imba. If you look at the leaf mesophyll cells, look at the arrangement of the mesophyll cells. Mesophyll cells sama khande ro desha. Look at the arrangement. So this is the special anatomy that we are talking about. C4 plant nalola special anatomy yore. C4 plant ki leaf nalola special anatomy yore le sambala tenda dila gorda. Yojun so bundle sheath cell yobache. Bundle sheath cell di khole che yore tuga yore. Layer manga yore. Ani di nyamtol khare yore chilola mesophyll cell di ge sangma. Bundle sheath cell gi ku chilola et ko thati buchen ko goso ne de do. So this is the difference. Now what do we call this particular arrangement of cells? This anatomy, special anatomy, is called as Kranz anatomy. You have to remember this word, Kranz anatomy. This particular arrangement of cells, bundle sheath cell che ani dik chilola ani kare kare mesophyll cell dige samo gogo sun de yosto the anatomy de langazo khalo Kranz anatomy law. Hope you're clear. Right now Kranz lana khalo gore Kranz law imena it is wreath. Kranz lana tsigi tunda de kare wreath law and wreath la du sambala khalo gore method de roj to gogan kyen roj mi de a medo so yo imena tsag so yo imena ye gogan kyen sto dil wreath la ani mi thong sa de la mangmi den zu gi khachi do kharsa khonju gi coffin gala den de ya method go go sheg do dile nga zu khala wreath la da inji gi tsigre. So the wreath Kranz anatomy law yumzen khalan look at the arrangement of the cells. Wreath nangshi reto. Ji meto gogo dinashi ro ani Kranz anatomy nalo la desida khonzu kharedo. Look at that. That is why it is called as Kranz anatomy. So I hope you are clear regarding that. Now let us talk a little bit more about the bundle sheath cells. Bundle sheath cells kan this kejam hans shewe imba na. If you try to find more information regarding that, the bundle sheath cells like kejo khare la they may be formed by several layers la so thanjing. Cell chig layer chig mai imba che cell mangbu gi layer in so yo sigres. Adin dal kejo khare la imba na. Bundle sheath cells, they have got large number of chloroplasts. For bundle sheath cells dige nang lola chloroplasts mangbu pair. And the main begi and cell walls, their cell walls are pretty thick. Cell wall dige pe thupu che. Cell wall thupu yo vimana khare it becomes impervious to gaseous exchange. Mesophyll cell gi chilo yo yogi ani air spaces nanglo ne lumbu phasu exchange ya yungumare. Kachin re la wimbena the bundle sheath cells, they have got thick walls and that makes them impervious to gaseous exchange. And konzu sama pe compact yor sida. If you look, all the cells, they are arranged in a very compact manner. Tightly packed redwa. Din zambala konzu parala intercellular spaces ke yo marde. No intercellular space. Intercellular space na khala ore cell ni paragi gap din sula. So there are no intercellular spaces. So this is the characteristics of bundle sheath cells. Bundle sheath cells na lo chloroplast mangbo yore. Walls thick yore di basonza, it becomes impervious to gaseous exchange. Lumbu yamatango mare. Then it also have a tight or compact arrangement which makes them have no intercellular spaces. Okay, so a difference in dini para la kheba de ha ku do. You can see the bundle sheath cells over here. Konzo gaji di kharado is chunga do ertaba do ni konzo kharisha. They have got direct connection with the intercellular spaces. So dini para kheba da C3 plant gi leaf da C4 plant gi leaf ni compare chewi imba na, you will easily identify C4 plant khandi me da C3 plant you khandi me ha ku. C4 plant ki example yakshu dikhare la wimba na magen gaji gi lomat khinna tir. Maize or corn is a C4 plant. So if you look at the cross section of a corn leaf, magen gi loma gaji cross section chi kiwe na vertical section chi kiwe imba na, you will clearly see all these vascular bundles over here. These are all vascular bundles. Major vascular bundle, minor vascular bundles. and you will see that they are covered by bundle sheath cells. And bundle sheath cells ki chilo la khardo mesophyll cell dike samo ko kora gyamne de do. So this is called as the Kranz anatomy, and this is the special leaf anatomy. Seuchung so C4 plants na lo. Tang ngazu ki di kana deshe khachin mangbu gyare. Kranz anatomy kana deshe mangbu khachin gyare lawim. In order to understand the C4 pathway, you need to understand the Kranz anatomy. Kranz anatomy na lo ni khari or laso. Kisi mesophyll cell di imbai na. If this is the mesophyll cell gi nanglo la ya khari ore bundle sheath cell yor. Again we will go back to this diagram. Mesophyll cell di imbai na. Mesophyll cell gi nanglo la khari ore bundle sheath cells yor. So ngazu gi mesophyll cell chikki and khoi gi immediate nyamdol yogyo gi bundle sheath cell chikki ni remove chikta wa imbai na. We will see this particular structure. Mesophyll cell chikche and bundle sheath cell chikki wa imbai na. You can see the structure over here. The di nanglo la mesophyll cell de to gang del to bundle sheath cell wa del to. Now you will see the C4 pathway de ha kur. Now atmospheric carbon dioxide jorwa. Atmospheric carbon dioxide di sugia nanglo lingre la wimbe na mesophyll cell ki lingre. Why? Because bundle sheath cells, they cannot take the carbon dioxide because their wall is impervious to gaseous exchange. Carbon dioxide direct lentu mar khachin re laimbe bundle sheath cells ki cell wall pe thupu yor laimba. Di indu zambala what will happen? The carbon dioxide cannot directly enter into the bundle sheath cell. So carbon dioxide khol zudo mesophyll cell na zudo. Carbon dioxide mesophyll cell na ya zubanda the carbon dioxide accept chinge de sure. C4 plants na la carbon dioxide primary acceptor de sure la wimba na phosphoenol pyruvate or short form na la PEP tinatir. Dinye de la gi primary carbon dioxide acceptor de suimbe RuBP imba. De la kharre C4 plant na la carbon dioxide acceptor di phosphoenol pyruvate re. So phosphoenol pyruvate is a 3-carbon compound. Carbon sumche yogi organic acid chigre. So carbon dioxide da and phosphoenol pyruvate nyamdo liya deni fixation, which is called as carbon dioxide fixation raw. So carbon dioxide fixation chungni, the first compound that is formed is a C4 acid ngela so dishin ngazu dila khala ore C4 pathway la lago jumze or thagarar. So phosphoenol pyruvate ki carbon dioxide nyamdo la ya jarwa da lamsa thangbu kharcha dure C4 acid cha dure di ming kha kona ti di khare oxaloacetic acid. Others you don't worry about that. First khare form chigre oxaloacetic acid form chigre. Okay, so oxaloacetic acid di ani acid shimbala gyuwatheni C4 acid shimbala gyuwatheni from malic acid to the aspartic acid chani, those acids they will go inside the bundle sheath cell through plasmodesmata. That yabu thonantia mesophyll cell the bundle sheath cell parala connection yorda nge dengazu jaimba cell wall kana jang duzambala. So plasmodesmata, they are like small holes through which there can be exchange of materials between the two cells, right? So C4 acid oxaloacetic acid the acid shimbala yuwathe raw and kharcha dure they will enter into the bundle sheath cell through plasmodesmata. Ta din nalo yogyo C4 acid di decarboxylate chini carbon decarboxylate chin na khala or kuma chani ani carbon dioxide chig nalo la le an ko kharcha dure C3 acid shadur, and this C3 acid will go outside, right, back to mesophyll cell. Bundle sheath cell ne mathweni mesophyll cell nalo to ani mesophyll cell nalo la C3 acid dikha jire, it will be converted back into phosphoenol pyruvate. That is called as regeneration. Phosphoenol pyruvate regenerate mache we imba ne ko zoru. So C3 acid cha yojoro dikha chi dure machilo lodoni phosphoenol pyruvate yangya suyur. So that is, it is called as regeneration. Now very important point you have to see over here. Decarboxylation kabla kharchi do dinalo carbon dioxide chig release chig do bundle sheath cell nalo C4 acid di khachi do carbon dioxide acid chig del release carbon dioxide release chig do. This carbon dioxide is used for Calvin cycle. Ta kharches Calvin cycle la di C3 plant nalo to yoy bache. C4 plant nalo le Calvin cycle la di yoy yorta. The difference is it happens only inside the bundle sheath cell. In C3 plants, it happens within the mesophyll cell. From there la mesophyll cell nalo la Calvin cycle la di chungu yomar. The Calvin cycle does not happen in the mesophyll cell over here. It happens within the bundle sheath cell. C4 plant nalo odi kejoyor. So Calvin cycle ki kharchi rani sugar suya. So you can see this is the main biosynthetic pathway. Biosynthetic pathway so the khari chadu gore Kelvin cycle chadu gore. Keju jigra ji khari loga yore la wimbe na atmospheric carbon dioxide ya nalo la fixed chewa da thangu khari form chikido. It forms a C4 acid which is called as oxaloacetic acid. The oxaloacetic acid the any ya nalo la doni chikido carbon dioxide ya nalo la ke yor ke ga du do. So carbon dioxide nalo la ke cha wa da ya rang ja chikido. C3 acid the phosphoenol pyruvate rang ja che ya carbon dioxide gone chile ya nalo ya ke ro chi yor. So this is the C4 pathway. I hope you are clear regarding this pathway. Okay, so dire it is also called as Hatch slag pathway. C4 pathway la ming shimba Hatch slag pathway because it was discovered. This particular this particular metabolic pathway was elucidated by Hatch and Slag. Hatch lani scientist cheda, Slag lani scientist cheda. Ni gi ani digala study cheyimba sonza pathway dilanga zu kharla gore Hatch slag pathway gel. Dinye kogo de langa zu kharla yo Kelvin cycle lago jumze khare because as Melvin Kelvin ki cheyimba cyclic cycle. Okay. You can see over here how it happens. Carbon dioxide, atmospheric carbon dioxide yana lo lewa da phosphoenol pyruvate ki accept che phosphoenol pyruvate acid kachi do yandi pep carboxylase ke yorda di para. This is an enzyme mediated reaction. Ani kharchi do oxaloacetic acid chag do. Oxaloacetic acid ne malic acid chag do. Malic acid yana lo do ni carbon dioxide ma le decarboxylation imba. Ani kharchi do it will turn back into pyruvate. Right? And pyruvate acid because C3 acid diya do ni ya kharchi do becomes phosphoenol pyruvate. Okay, so this is the C4 pathway. The main carbon dioxide ya nalo la leni carbohydrate swing so cycle Kelvin cycle. So Kelvin cycle is common in all the plants. Okay, now to know one more difference between C3 plants and C4 plants, photorespiration C3 plant nalo la khar chungore photorespiration chungore. C4 plant nalo la photorespiration chungo marta. Photorespiration is a wasteful process. Photorespiration it is a wasteful process and C3 plant nalo photorespiration la chungore C4 plant nalo photorespiration la chungo mar. So photorespiration la khari du tainda. Photorespiration la di khari imba gotswala you need to know the nature of RuBisCo enzyme. Right? RuBisCo la khari imbe Ribulose bisphosphate carboxylase oxygenase la imba. So carboxylase oxygenase this particular enzyme can bind with both carbon dioxide as well as oxygen. The enzyme jorwa. Active site kamada near. Chikare la wimbana carbon dioxide kala jareg nubore. Niva the kharela women oxygen le jareg nubayor. The Hindu zambala carbon dioxide la jarwa imbana it will bind that carbon dioxide along with RuBP nyamdola jani. RuBP nyamdola Ribulose bisphosphate nyamdola jani ani 3-phosphoglyceric acid sugyo. But if this particular enzyme RuBisCo enzyme la the oxygen nyamdola jarwa imbaina what will happen? Right, oxygen nyamdola jarwa imbaina it will form phosphoglyceric acid, phosphoglycolate plus carbon dioxide. This is RuBisCo the oxygen nyamdola jarwa imbaina kharchire phosphoglyceric acid ani phosphoglycolate and carbon dioxide din nanglola ani sugar ge swetugumare NADPH ge swetugumare ATP ge swetugumare mogo kharchigre they will use them, right? So therefore it is a wasteful process. Sugar swematu vimena khachigre the reaction dikharchare it's a wasteful process char tigal thandu khachigore ATP to son rugor. So it is a wasteful process. Now this particular reaction called photorespiration, it happens within the C3 plant. C3 plant now photorespiration chungor. Whereas in the C4 plants, photorespiration does not happen. Photorespiration chungomar C4 plant now. Di machungu gumzen kharla we mena ngazu gi remudila ta warda. Di nanglola mesophyll cell di. Mesophyll cell nanglola RuBisCo enzyme la di yomar RuBisCo enzyme chorwa RuBisCo enzyme di mesophyll cell na yomar so carbon dioxide nanglo ya lewa da ani de danglo la ani oxygen nyamdo la jarni RuBisCo enzyme de le medu zambal RuBisCo enzyme di khwale yore di nanglo yor so RuBisCo enzyme is located within the bundle sheath cell. So bundle sheath cell nanglo oxygen ligo mar khari ligo ore bundle sheath cell nanglo la carbon dioxide chigo ya ke do the C4 acid digi oxaloacetic acid the malic acid ya nanglo do ni carbon dioxide ya nanglo ke shek do. So what happens the nanglo la carbon dioxide ki concentration manga che oxygen nyunga yor in du zambala RuBisCo enzyme di nanglo yo di khatu ne kharinyam do la bind chigre carbon dioxide nyam do la bind chigre and photorespiration like process the chungo mar. Whereas in C3 plant nanglo la khonzo nanglo la photo kharsa manzo ki photosynthesis ki dark reactions sama khwala chungo re ro Calvin cycle dige sama khwa chungo mesophyll cell da dige na chungo ro di chungdu zamba la harchi gore inside the mesophyll cell they have got RuBisCo enzyme khonzo ki mesophyll cell nanglo RuBisCo enzyme diyor de ki mesophyll cell nanglo RuBisCo enzyme yo mare la ba C4 plant nanglo la ro mesophyll cell la RuBisCo enzyme yo mares RuBisCo enzyme di khwa na zu de re bundle sheath cell nanglo yoro de la khari yore RuBisCo enzyme di mesophyll cell nanglo yor di indu zamba la carbon dioxide da oxygen ki concentration kharchi dure almost chibado cha degrit nanglo la ro di indu zamba la what will happen there is a competition between carbon dioxide and oxygen for binding with the RuBisCo enzyme la jara ki thapshi chi ro carbon dioxide oxygen ni ga ro. So what will happen sometimes oxygen will bind with RuBisCo enzyme and that will lead to the wasteful process called as photorespiration la de chungor. So photorespiration la de chungdu zamba la ni kharalas it is a wasteful process la so energy to son thewa ro sugar game so. So what will happen it will be khare detrimental for the C3 plants this chongra warta ro C4 plant la khepsan yor di du zamba la C4 plant di productivity yaga yor la go jumze wartan chi thakara re C4 plant di C3 plant nyam do compare chew imba na productivity yoga yor la akiran zu samta imba na de da magen ni durbo imba na magen go shu sarlon che du do ro namshi chaw yo imba na ko sarlon go shu che do chunyu nyu ne sarlon go be che du do di che to go jumze khare la imba na C4 plant magen nang lo la photorespiration la di chungo mar di du zamba la ko kharchi gore growth ki kyoga yor productivity kyoga yor. So these are the differences between C3 plants and C4 plants. Right. Now, kiranzo assignment chiketa. Dikhare try to complete table 13.1. 13 point table kharsa 13.1 kiranzo lotem nal te shawadi complete chetu wi mana the table dige sama jang tu wi mana then you will know whether you have understood the differences between C3 plant and C4 plant. Unyi ni paragi khebadi kiranzo gotsoye me de takje khande chetu ro la wi mana lotem nalo yo table 13.1 di kiranzo solve che try chida. So diche in the next video, we are going to discuss about factors that affect photosynthesis. So please read about factors affecting photosynthesis part, and then you can watch the next video. Okay, thank you.